Hey everyone, today I would like to give you some background about the chlorophyll fluorescence data that you'll be working with in class. What we're going to be talking about today are the photosynthetic light reactions, also known as the photosynthetic electron transport chain. And so during photosynthesis, chlorophyll absorbs light energy. That light energy is transferred to what's a special chlorophyll, which is known as the reaction center. And in the reaction center, what happens is electrons are taken from water, and they are ultimately used, those electrons are ultimately used to reduce the molecule NADP to NADPH. And so this reduced molecule, NADPH, provides the reducing power needed later on in the carbon reactions of photosynthesis. At the same time, hydrogen ions are pumped across the thylakoid membrane, which builds up a proton gradient, and this proton gradient is used to drive the ATP synthase and create ATP, and that ATP is also later used during the carbon reactions of photosynthesis. And we'd like to be able to, as plant biologists, measure some of what's going on during these light reactions. To do that, we can take advantage of the fact that not all of the light that is absorbed in this reaction center actually goes towards photosynthesis. Some of it is released through other processes. And so we'll get into that in a few minutes. But first, let's look at the same diagram of the light reactions, but shown in terms of energy. And so here what we have is exactly the same diagram, but instead of showing it based on like a cartoon of the various protein complexes within the membrane, we're showing it in terms of the energy of each step. And so what we start with is this reaction center, the photosystem 2 reaction center, which is known as the P680 chlorophyll. And it's in a relaxed state, which we also call the open state. And when it absorbs light, it goes into this excited state, a higher energy state, and we can call that P680 star, or the closed state. So when P680, this chlorophyll reaction center in photosystem 2, is in the P680 star state, it cannot absorb any more light. And then those electrons are transferred through various other electron carriers until it gets down to this photosystem 1 reaction center, where more light is absorbed, and then that re-excites it, and you get the photosynthetic electron, the center of the photosystem 1 becomes excited, and it will be P700 star. So the difference between P680 is the chlorophyll reaction center for photosystem 2, and P700 is the chlorophyll reaction center for photosystem 1. And we're going to focus most of our attention on this first reaction center in the process, which is the photosystem 2 reaction center, P680. And so we would like to know, of all the light that is hitting the plant and being absorbed by the plant, how much of it is going to this process of photosynthetic electron transport, and how much of it is going to other processes. So if you think about all of the light that is being absorbed by the chlorophyll in the plant as water in a barrel, there are several possibilities for that, where that water can go. So we can think of those as different holes in the barrel. So one of them, big hole, is photochemistry, and that's the process that I just briefly described. That's the photosynthetic electron transport, where electrons are used to reduce NADP to NADPH, and protons are in the process pumped across the membrane, and later that proton gradient is used to create ATP, and then the NADPH and the ATP are used to fix inorganic carbon into organic carbon during the carbon reactions of photosynthesis. So that process is called photochemistry. Another hole in the barrel is what's known as non-photochemical quenching. And so that's what happens when the chlorophyll reaction center of P680 absorbs light, goes into the P680 star state. So it's an excited state, but for whatever reason, photochemistry can't operate fast enough to use up all of that energy. And P680 star is very unstable, and so the energy will be dissipated in some way. And in order to prevent that energy from being dissipated in an uncontrolled fashion, plants have several processes that they can use to dissipate the energy in a more controlled way. And we can lump those together and call them non-photochemical quenching. So basically, the excited state of the chlorophyll is quenched and not through photochemistry, so we call it non-photochemical quenching. And then finally, there's a small amount of the light that's absorbed that's immediately, that light energy is dissipated through immediate emission of a new photon. So this is fluorescence, and it happens at a rate that's proportional to the rates that are happening for these other processes. And the thing about fluorescence is because of photons actually being emitted, we can measure it. So you take a spectrophotometer and you place it on the leaf as a detector, and you can actually measure those photons that are being emitted directly. And it's not easy to measure any of these other processes directly. And so we have a device or different devices that you can measure the fluorescence, and we'd like to use information about the rate of fluorescence to determine how much of the light is going on in these other biological processes. And so that's what we're going to do when we calculate various chlorophyll fluorescence parameters, are basically using this information from chlorophyll fluorescence that we've measured to calculate what's going on with these other processes in the leaf. And so what we can do is a little experiment. And so this is a diagram of the typical experiment. So first you take a leaf that's in the dark, and it has some level of fluorescence. We can call this F0 or F0. And we'll turn on a measuring beam. So the measuring beam is a very low level of light. It's so low that it is not bright enough to trigger photosynthesis, just bright enough in order for us to actually pulse the reaction center with a bit of light and see how much light is re-emitted based on fluorescence. So we can measure the fluorescence when the leaf is in the dark, and if it's been in the dark for a sufficient amount of time, all of the photosystem 2 reaction centers should be in their open or relaxed state, so that P680 as opposed to the P680 star. Okay, so after we've measured this initial level of fluorescence in the dark, we can hit the plant with a saturating pulse of light. So this is a really extremely bright light. And so we give it a pulse of an extremely bright light that's so bright that all of the photosystem 2 reaction centers will become closed or be in their excited states, the P680 star state. And we can measure the fluorescence again right after this saturating pulse of light. And we'll call that FM naught, or maximal fluorescence, because again, fluorescence is proportional to the other processes that are going on. So when we hit it with a saturating pulse of light, and you have more light, there's more fluorescence. Okay, so we have more fluorescence. This is the max, and in this case, it will be the maximal amount of fluorescence that you can theoretically get, because all of the chlorophyll holes in the photosystem 2 reaction center will be in their excited state. Okay, and the reason we do this in the dark is to ensure that, or a dark adapted leaf, a leaf that's been in the dark, is to ensure that all of the photosystem 2 reaction centers were in the open state, relaxed state to begin with, and there was no non-photochemical quenching going on at the time, because the leaf was completely relaxed. Okay, so after we've measured both the initial fluorescence in the dark and this maximal fluorescence, then we turn on what's called an actinic light. So this is a light that is bright enough to trigger photosynthesis. And then we can measure fluorescence over time. And what you'll see originally when we turn it on, you'll see a lot of fluorescence, and it slowly goes down. And that's because this other process of non-photochemical quenching turns on, and that is going to dissipate some of the light energy. So after some period of time, what we can do is hit the plant with another saturating pulse, and then we'll measure fluorescence again. And the fluorescence after the second saturating pulse, we'll call that FM, again for maximal fluorescence, but prime, because this is a plant that's been in the light, and so it has some level of this non-photochemical quenching process going on. Then finally, what we'll do is we'll turn off that actinic light, so the leaf will now be in the dark again, and we can measure fluorescence again, and that will be F naught prime again. So F naught, because this is the minimal fluorescence, but after the plant has been light adapted, as opposed to the original F naught parameter, which was in the dark. And then finally, we can measure this FT parameter. So that's the fluorescence at the end of the experiment, right before this saturating pulse. And so from these various fluorescence parameters, we can actually learn a lot about what's going on in the leaf. So one thing we can do is measure the maximal yield of photosystem 2. So this is the maximal amount of light energy that can be going towards photosynthesis. It's a parameter that's called FV over FM. FV stands for the variable fluorescence, and it's just the difference between this maximal fluorescence and the minimum fluorescence. So the maximal fluorescence right after a saturating pulse for a dark adapted leaf, and the F naught, the minimal fluorescence, before any saturating pulse in the dark for a dark adapted leaf. So F naught is the fluorescence when all of the reaction centers are open. FM is the fluorescence when all of the reaction centers are closed. So the difference between those is the variable fluorescence, and you just divide that by the maximal fluorescence, and that gives us a parameter called FV over FM. And this is used a lot to measure sort of the health of a plant, because it tells us how much of the light energy, what is the yield of photosystem 2. So what is the yield for photosynthesis? Another parameter that we can measure that's somewhat related is called phi PS2, and this is also known as the efficiency of photosystem 2, because it measures the proportion of the light energy that is going to drive photochemistry. And the way that we calculate this was actually all based on light-adapted measurements. And that's, we take the FM prime measurement. So that's the maximal fluorescence after saturating pulse, but in a light-adapted leaf. And we subtract it from, instead of the minimal fluorescence in a dark-adapted leaf, the fluorescence at the end of this experiment after we left the light on for a while. And so that's called FT, and then we just divide it again by FM prime. And that gives us the efficiency, so to speak, of photosystem 2. So it's will be a fraction between 0 and 1, where the higher that number is, the more of the light energy is going to drive photochemistry. And then we can actually also, so this is just a proportion, but if we want to know more in absolute terms how much photosynthesis is happening, we can calculate a third parameter that we call J, which is a measure of linear electron flow, so how much photosynthesis is actually happening. And this one is actually very hard to calculate exactly, but we can get an estimate of it. So the J is equal to the phi PS2, so this efficiency of photosystem 2, which is just a proportion of the light energy absorbed by the chlorophylls that are going to photochemistry. And then we just multiply that by the total absorbed light. So this PFDA stands for photon flux density absorbed. So this is the light that's absorbed by the leaf. And then we just multiply that by 0.5 because in this calculation we're assuming that half of the light energy is absorbed by photosystem 2 and half of it's absorbed by photosystem 1. So you can already see there are a couple assumptions in this calculation. One, that the light is equally distributed, and that may or may not be true. And the other problem is that it's very hard to actually measure the exact amount of light that's absorbed by the leaf. And so often we end up estimating this as a relative term based, instead of on the light absorbed by the leaf, based on the light hitting the leaf, which we can measure with a light meter. But in any case, this parameter gives us a little bit of a sense about how much photosynthesis is actually taking place. So the efficiency of photosynthesis, what proportion of the light is going towards photochemistry, is measured by phi PS2. But the total amount of linear electron flow, so the total amount of electron transport, is measured by this J parameter. And then finally we can measure non-photochemical quenching, which is just the proportion of the light going to these other processes that the plant uses to dissipate the light as opposed to photochemistry. And the way we do that is we start with the difference in maximal fluorescence between the dark adapted state, where we're assuming that there's no non-photochemical quenching. So that was this FM prime parameter. And we subtract from that the maximal fluorescence after we've had the leaf in light, so the F prime maximal fluorescence, and then we just divide that again by this FM prime. And that will give us a sense of the proportion of light that's going towards non-photochemical quenching. And so you can see that, in the dark adapted leaf, you have, when you give it a saturating pulse, you get a big spike in fluorescence. And when you give it the same saturating pulse after the leaf has been exposed to the light, you get a smaller spike in fluorescence, and the difference is proportional to the amount of light energy that's getting dissipated through this process of non-photochemical quenching. Okay, well, I hope that helped. in giving you a little bit of background about what some of these chlorophyll fluorescence parameters are. And if you'd like to discuss this further, I would be happy to do that.